Hello, I'm Debora Angeloni, Associate Professor of Molecular Biology at Sant'Anna. My group and I work in the field of space biology, and that is what I'm going to talk to you about today. I wish to start this journey from a sentence. There is no land uninhabitable, nor sea innavigable. It is from a letter by Robert Thorne, an explorer and cartographer, that in 1527 was asking the King Henry VIII to support new ocean explorations. This old sentence is relevant for explorers of all time, and certainly today for space explorers. Because just like exploring the oceans was dangerous back then, going into space nowadays is very much difficult, very expensive and dangerous. For example, in going to Mars, a human crew will be exposed to dangerous radiation, altered gravity fields, a closed environment with isolation and confinement issues, aboard spaceships for a long journey at a far-growing distance from the Earth. So why would we humans want to go into space? If we ask different people this question, for example, an economist, a politician, a scientist, we might hear different answers. In fact, some look at space as an opportunity for business. Others look at it as an opportunity to widen their national area of influence or defense. For scientists, curiosity, the inclination to explore, and the ability to adapt to new environments are characteristics as old as humans. Since a few years, our societies have been considering in a realistic way the possibility of living and working on celestial bodies other than the Earth. But we need to know more about what happens in space. Today, the solar system is being explored with satellites and rovers. However, if humans want to extend their presence in space, we need to send humans up there. Because human explorers confer the flexibility and adaptability to different scenarios that are impossible to achieve in robotic missions. And so, major space agencies of the world, including the European Space Agency and the Italian Space Agency, are concretely planning long-duration missions that will involve astronauts living and working for a prolonged time outside the Earth. This is why we need space biology. Space biologists study how spaceflight affects living beings to increase our basic scientific knowledge and to contribute to guarantee health and safety for human crews with safe and productive work environments during missions and with rehabilitation afterwards. Since the beginning of the space age, the effects of space flights were considered unpredictable and even dangerous, to the point of fear the disintegration of bodies. So, space explorations require some serious biological and biomedical research. Just 1 month after the launch of the first satellite, the Sputnik 1, in October 1957, the Sputnik 2 was launched with the first living being on board, and it was a dog, Laika. Then, in 1961, 2 humans followed, Yuri Gagarin in April and Alan Shepard in May. Why is it so important to know the effect of microgravity on the human bodies? Well, when we talk about space, we actually mean spacecrafts, that is special closed-circuit artificial microcosms where everything from the atmosphere to the water is built, recycled, or imported with periodic launches from ground. In these environments, many parameters differ from the Earth, and each of them, deviating from normal, can represent a risk factor for life. The major risks of space flights include hypoxia, that is lack of oxygen, accelerations, decelerations, extreme excursions in temperature and pressure, and variations in circadian rhythms, as well as psychological factors deriving from isolation and confinement, exposure to cosmic radiation, and microgravity. All these factors had already been the subject of scientific and medical research before the space age. All but one, microgravity, which is probably the most peculiar parameter of space flights. We need to know how living matter reacts to this altered force. Just think of the way free water flows in space, as we have seen several times on TV from the International Space Station. You can imagine that something peculiar happens in all our cells. Each cell can be represented as a bag filled with a highly viscous liquid. Now, current research aims to characterize the independent or synergistic effects on complex phenomena such as DNA damage repair, immune response, metabolism, bone and muscle homeostasis, and behavior during a space flight. And here, space biology comes to action. Space biology has several fundamental objectives. For example, understanding which molecular, biochemical, or genetic mechanisms of the cell are affected by spaceflight. Identifying the molecular processes activated in response to this complex stimulation. Exploring the effects exerted at a more complex level than cells, that is, tissues, organs, and entire organisms. Elucidating if and how space influences basic biological processes to the point of causing dysfunction and disease. So what are space biologists interested in? Space biologists study human crews, but also different experimental models, including other animals, plants, and microorganisms. The long-term goal of space biology is to contribute to the sustainability of living and working in space, as well as to promote health and well-being on Earth. So space biology asks a few fundamental questions. For example, what can we learn from observing the response of living organisms to microgravity? And this is a question of basic research. Or how can we better protect human health in space? And these are questions of applied research. So it is important to stress that whatever it's been discovered or invented to support space research, it has also been applied to life on Earth. And that is true for technological products as well as for biomedical research. There are illustrious precedents in history. For example, in 1768, James Cook set sail on the first of three important famous exploration voyages to the South Seas. In order to prevent scurvy, Captain Cook decided to carry on board a large amount of truly expensive fresh lemons and other vegetables, because he had decided to trust the theory of James Lind, a young doctor that had published a book a few years before titled A Treatise on the Scurvy. That was in 1753. The scurvy back then was really a terrible and mysterious disease that accompanied the ocean explorers in their voyages. And in fact, it could kill a great many of sailors during these long voyages. So following this intuition, Cook did not lose a single man to scurvy. And in time, the recommendations of Dr. Lind were extended to the civilian population. So what happens to the human body in space? We know that all organs and organ systems are affected, to the point that astronauts become a model themselves for studies on aging. And in fact, the human body is not programmed to live in the absence of gravity, and it enters a catabolic, destructive mode. Tissues like muscles and bones become sort of useless because they are used in a different ways than on ground, and so they are literally destroyed. Research on astronauts has shown that microgravity affects multiple physiological systems, especially fluid distribution, the cardiovascular system, and muscle skeletal systems. In space, microgravity produces a combination of factors that cause an astronaut the same kind of deficit experienced on Earth by persons with physical handicaps. And therefore, the study of motor, visual motor, and perceptual deficits experienced by astronauts provide researchers with important information on possible compensation strategies useful for people affected by pathophysiological deficits on ground. In 2019, we learned about the results of a large study that NASA, the National Aeronautics and Space Administration of the United States, conducted with a focus on characterizing human adaptation to space. They engaged two identical twin astronauts. One was sent in space for 342 days, and the other remained on Earth. And by the end of the study, NASA compared the astronauts' health and performance. The study lasted 25 months, during which many data were gathered before, during, and after the space mission. And really an impressive number of tests was performed on these individuals. And the results showed us that indeed all organs are affected by spaceflight. However, given that the majority of biological and health variables remain stable or return to baseline shortly after the 340-day space mission, the study suggests that human health can be sustained over this duration of time of spaceflight. Of course, we do not know about the effects of missions longer than 1 year, and those should be studied and measured in future astronauts. But why is the force of gravity so important for living beings? Well, we must remember that gravity is a force with fixed intensity and direction that has been there since the dawn of life on Earth. It has been a fundamental force in accompanying the evolution and probably has played a great contribution in determining the forms and functions of life as we know it today. For example, the viscosity of cytoplasm, that is the content of our cells, the positioning of organelles within the cells, and the organization of the cytoskeleton, that is the structure that allows the cells to keep a shape and interact with surrounding environment, were all probably engineered by evolution as countermeasures to the force of gravity. So in space, where gravity is a millionth of the ground value, all our cells respond to and adapt to this unprecedented situation. Space biologists are interested in studying microorganisms, especially the pathogens, because in spaceflight their proliferation rate, their virulence and drug resistance do change in significant ways. And so even moderate pathogens could become important risk factors for human crews. Special attention in recent time has been devoted also to the human microbiome, that is the sum of all microorganisms that colonize our body from within and on the surface, because they are extremely important to determine our health. The microbiome changes in response to environmental conditions, and now it is known that it changes also in spaceflight. Also, plants have been studied. Plants have been privileged travelers of spacecrafts because they do perform a fundamental function within every possible ecosystem compatible with human presence. That is, they produce oxygen and energy through chlorophyll synthesis. Plants can also represent a source of fresh food, and finally, but not least on the scale of importance, they have a positive effect on the psychological well-being of space crews. So over time, different plants flew into space thanks to various experimental greenhouses. And in 2015, the greenhouse veggie by NASA allowed astronauts to consume fresh vegetables entirely generated in space. Plants use environmental references to grow, and two of these, light and gravity, play a substantial role in directing the oriented growth of the roots and the foliar apparatus. And what about animals? Well, a large variety of species has flown in space, depending on the specific experimental questions to answer. Different species were studied to investigate changes of gene expression at organ level, embryogenesis, or different physiological responses regarding, for example, muscle atrophy, bone loss, impairment of microcirculation, and so on, up to behavior. As cell biologists, my group and I were interested in knowing more about the effects of spaceflight on a specific type of cell, that is the endothelial cell. This is a cell type that lines the inner part of all our blood and lymphatic vessels. There are differences in the specific characteristics of endothelial cells from a district of the body to another and from one type of vessel to another. And yet the sum of all these cells makes up an impressive number and constitute an organ called the endothelium that plays a fundamental role in maintaining the equilibrium of our body with the environment, a situation that is called homeostasis. And in fact, these cells perceive signals from the outer world and send signals on their own. And when this complex system is deranged, some important pathologies can arise. We were interested in this specific cell type because many astronauts on the return from space reported problems that can be attributed to malfunctioning of the endothelium. With the support of the Italian Space Agency and the European Space Agency, we had the opportunity to send to space human live endothelial cells and to study and observe their response to spaceflight. My colleague Ivana Barravecchia will tell us more about the methods that we use to study the material upon its return from space. Please, Ivana.
For our experiment, we assembled 40 microincubators at Baikonur Cosmodrome. 24 were sent to International Space Station. When the cells come back to ESA, we analyzed the changes induced by spaceflight. In particular, we studied the gene expression changes with advanced sequencing technique, like the next generation sequencing. With this technique, we observed the gene expression changes induced by the spaceflight. The second stuff that we analyzed was the cell shape, morphology, and volume. In particular, we analyzed this structure thanks to staining techniques that help us to visualize better under the microscope cellular and subcellular structure. You can observe the most important alteration in cytoskeleton protein. Cytoskeleton is an important structure in cells and gives cells mechanical structure, is involved in movement in internal organization. In particular, you can observe three important proteins present in the cytoskeleton: F-actin, vimentin, and tubulin. F-actin is the filament of fibrillar actin, and you can observe how in space flight is completely dissolved, while in control cells you can observe this filament structure inside the cytoskeleton. The vimentin, there is a protein of intermediate filaments, usually are accumulated in the perinuclear area. In the spaceflight cells, you can observe that the vimentin are more relaxed inside the cells. The same happened for tubulin protein that are more relaxed inside the cytoskeleton.
Thank you, Ivana. And let's move on with the results that we got from these studies. We discovered that microgravity and space radiation exert opposite effects on these cells, on many of the molecular signal pathways that are acting on these cells. And this is something that was never heard of before. Microgravity would have activated a self-help mechanism called autophagy, whereas space radiation inhibited it. And more important, this observation suggests a possible target for a pharmacological intervention to ameliorate the endothelial cell suffering that we mentioned before. Is all this relevant to us on Earth? Well, the answer is yes. because just like it happened for the sailors of Cook's crew, the results of space research could be used to help ameliorate problems related to human aging, physiological aging here on Earth. As we know that, for example, aging causes an inhibition of cell autophagy. Is there a way to reproduce the absence of gravity on Earth? Well, actually, the answer is no, there isn't, except for very small objects like seeds, insects, or very small plants that can be accommodated on simulators of microgravity, like the rotating wall vessels or the random positioning machine. For larger objects like animals or humans, there are special flights that are realized with a zero-G airplane. The airplane performs a phase of horizontal flight and then starts a very deep ascent, and at the peak of the parabola, pilots perform a steep descent phase, at the end of which they start flying horizontal again. At the peak of the parabola, the airplane's content experiences a few seconds of microgravity, and usually the airplane realizes about 30 parabolas in a sequence. But this does not last long enough to allow long-term observations of biological systems. And so in respect to this problem, a space station like the International Space Station is truly a priceless facility. The other side of microgravity is represented by hypergravity, that is an increase of the force that we can obtain in a very controlled way in centrifuges designed for scientific materials or even for humans. From our studies, we noticed that the cells that we sent to space and respond to microgravity or hypergravity in quite the opposite way, at least for some cell biomarkers, that is, some components of the cell machinery that we used as landmarks to compare the different experimental treatments. And now we are working to understand whether hypergravity can be used to ameliorate some problems associated with aging, based on our observation that aging is associated with some structural changes similar to those exerted by microgravity in spaceflight. What are the major challenges of current space biology? Certainly areas that are essential for life support. So systems for water and air renewal and waste recycling, but also protection from radio exposure, countermeasures to microgravity, maintenance of human performance with an attention to prevention of behavior problems in a closed and confined environment, as well as on-site and remote control medical care. The study of innovative methods will accompany these advances and will help us on Earth, for example, artificial gravity or hypergravity produced on board spacecraft as countermeasures to the effects of microgravity could also find therapeutic applications on Earth. An interesting subject is represented by synthetic torpor, which is an experimental analog of hibernation that could be applied to overcome the consumption of resources and the psychological and physical stress on long journeys. Although it looks futuristic now because humans are not hibernating animals, this is the subject of active current research and could represent a game changer for long-duration space missions. Anyway, the progressive solutions to the biological challenges represented by the status of human beings in space will offer fundamental advances to our ability to explore and therefore to know more. Hopefully, this will also contribute with advanced solutions to the current environmental challenges, because as of now, this planet is very obviously the only one that we have for living.